Okay, now we're recording, so we'll go ahead and get started on the actual lecture. So let me share PowerPoint with you guys here. There it is. Okay. All right, so these slides, of course, I will post on D2L, so you'll have access to all of the slides. So you can make sure to take a look at those. And then I will also post, if you want to revisit the lecture, this recording will be posted on D2L as well. So you can take a look at that if you want to revisit any slides that we talk about. So today what we're talking about is molecular biology. We're going to kind of be talking about the processes of the cell and how we go from the information that's stored in DNA to the information that the cells need to do their cellular functions. So that is basically referred to as the central dogma of molecular biology. So a dogma, just in general, is this set of beliefs that people agree upon. So when we talk about the central dogma of molecular biology, we're talking about how biological molecules use the information that's stored in DNA in the cell. So there are 3 steps involved in this. Basically, we're going from DNA to RNA to proteins. And so we're going to talk about the molecules involved in these and how we go from DNA to RNA to proteins. So again, these 3 molecules, they're right here. They have different functions. So our DNA is responsible for storing information. So we've talked about DNA a little bit already, but this is our double-stranded molecule, and we're going to break down the structure of this here in a few slides in a little bit more detail than we've done in past lectures. But in addition to DNA, we have RNA. So this is also a nucleic acid. So we're still using this nucleic acid macromolecule, but instead of having a double-stranded piece of nucleic acid, we have a single-stranded piece of nucleic acid. So this isn't necessarily storing information, but it's just transporting information from DNA to a different part of the cell. And then we also have our proteins. These are what are going to be actually performing the functions of the cells. And we've talked about proteins a lot. Specifically, we've talked about lots of different kinds of enzymes and things like that. So these are going to be how we're performing the functions in the cell, and these are going to be created by our RNA molecule. So we have DNA that's transformed into RNA, and then RNA is turned into proteins. So the 3 steps that we have with these 3 molecules, we have the option of DNA replication. That's the first step. We've talked about DNA replication when we talked about mitosis and the cell cycle and meiosis. This is, remember, occurring in that interphase section of the cell cycle. This is where we're just turning DNA into more DNA. This is an exact copy of the DNA. Then when we're going from DNA to RNA, that's called transcription. So we're transcribing one nucleic acid into a different type of nucleic acid. And then the third step is translation. And this is where we're going from our RNA to our protein. So we're translating information from one macromolecule into another macromolecule, going from that RNA to that protein. And then we have different enzymes that are involved in this process. We're going to add a few more enzymes to this list, but these are the main ones that are involved in building these macromolecules, these chains of nucleic acid or chains of amino acids. So DNA replication is going to be using DNA polymerase. So polymerase just referring to adding more. Oh, Jaden, you can't hear me? Can everybody else in the chat, if you can hear me, make sure you let me know. Am I on mute right now? I think I'm still talking. Okay. Can you guys hear me? Okay, Baylor can hear me. Jaden, you can't hear me? All right, I think a few other people can hear me, so it looks like, I don't know what's going on your end, Jaden, but I can definitely, it says on my end that I'm not on mute and other people can hear me, so hopefully if you want to like exit the meeting and come back in, maybe that would help reset it. But it seems like other people can hear me, so I'm just going to keep going and hopefully you can chime back in, Jaden. Okay, so DNA replication. Oh, okay, hold on. So give me a second, guys. My boyfriend's helping me with WebEx because he's better at this than I am. If you're having trouble hearing me, you can call in. How do they do that? Okay, so there's a number in the room. Like when you click on the link to join the room, there's a phone number and you can call in with your phone. That might help for the audio if you're having trouble with audio. Thanks, Carl. Okay, so DNA replication, that is DNA polymerase. Transcription is going to be RNA polymerase. So again, polymerase is just where you are adding more polymers to the molecule, right? So we're adding more nucleic acids to RNA with RNA polymerase. And then for translation, we're using something that's different. It's not really an enzyme necessarily, but we're using the ribosome. So we talked about the ribosome when we talked about all of our cell organelles. So the ribosome is going to be one of those organelles, and this is involved in building those proteins. So it's basically like a protein polymerase, right? We're adding more amino acids to our protein, increasing the length of that polymer. So that's what we're going to be using to build that protein. Okay, so when we're in DNA replication, we are creating two identical copies of that chromosome. So these are going to be identical copies, DNA to DNA. This is only happening during interphase, right? During that S phase, that synthesis phase. And we're only doing this before we divide, right? We don't want to make a big copy of our DNA if we're not going to divide that DNA in half again. So we want to make sure that we're doing this prior to cell division, but we don't have to divide in order to continue with the rest of this, like the transcription and translation, right? We don't have to replicate our DNA and divide if we want to turn our DNA into RNA and our RNA into proteins. But this is sort of part of this central dogma, so I want to make sure we touch base on it and how it happens, because it is pretty similar to the transcription process, but we don't have to replicate before we do transcription or translation. Okay, so let's talk a little bit about the structure of DNA before we talk about the replication of DNA. So the structure of DNA, this double helix structure, was discovered back in the 1950s. There are two scientists that are credited with discovering this DNA structure. They're Watson and Crick. They won the Nobel Prize in, I think it was 1962 or 63, for their discovery of DNA structure. So they pretty much discovered this structure based on this photo here. This is an X-ray photo of a DNA molecule. And they took all of their information they knew about the structure of the components of DNA along with this X-ray photo and determined that it was a double helix. But this photo was actually taken by a scientist named Rosalind Franklin. She worked at a different institution. She worked, I believe it was at an institution in England, and Watson and Crick were in the US. And there was a scientist that she was colleagues with that took this photo without her permission and gave it to Watson and Crick. And then they got credit for discovering the structure of DNA when she actually came to the exact same conclusion. So she is really the one who deserves lots of the credit, and she doesn't get a lot of the credit. So there's a link to a video here. We're not going to watch it right now, but I am going to post it in the lecture material for chapter 9. I would recommend you watch it because she really kind of missed out on getting all of the credit. Watson and Crick decided to share their Nobel Prize with the person that stole this photo and gave it to them, but they didn't give her any credit. So I would definitely recommend taking a look at it because she is a pretty fascinating scientist and was a pretty incredible thing for a woman to do back in the 1950s. So go ahead and take a look at that video if you've got a couple minutes. So within the DNA structure, these building blocks are nucleotides, right? And they consist of 3 different parts. So there is a sugar, a deoxyribose sugar, there is a phosphate group, and then there is a nitrogenous base. And these sugars and these phosphates make up the backbone of this DNA molecule, and then the bases are really important because that's sort of how these 2 strands are staying together, right, in this double helical structure. Okay, so the sugar, I want you to make sure that you know it's a deoxyribose sugar. That's important because that's sort of why DNA is named DNA, deoxyribonucleic acid. And this sugar is different in RNA, so we want to make sure we know that DNA has the deoxyribose sugar. So there are 4 types of nitrogenous bases within our nucleic acid. You're going to need to know the names of all 4 of these, and you're going to need to know which type of nitrogenous base they are. So we have adenine and guanine. Those are often just shortened to A and G. And these are purines. These have 2 rings. These are a 2-ringed structure. You can see that there in the image. And then cytosine and thymine, or C and T, are the pyrimidines, and they have a single ring. And it's important we know that the purines have the 2 rings and the pyrimidines have the 1 ring, because the way that these nitrogenous bases interact, a purine is always going to interact with a pyrimidine. A double ring is always going to be interacting and bonding with a single ringed structure. We're never going to have 2 single ringed structures bonding or 2 double ringed structures bonding, because then the length in between our 2 backbones of DNA would be a different size. We want it to be like a ladder, like each rung of the ladder, we need to be the same length. If there was a rung of the ladder that was shorter or a rung of the ladder that was longer, that would make for a very strange ladder, right? So we want those to all be the same length. So our double rings are always going to be pairing with our single rings. And our nucleotides are named by which base they have. So that whole nucleotide, that sugar, that phosphate, and this base are referred to as an adenine or a guanine. So that whole nucleotide is named by these bases because that's how they differ. So that's how we can tell them apart. So as I mentioned, the sugar, the deoxyribose, and the phosphate are forming the backbone. So they're forming this structure right here, this helical backbone. And then the nitrogenous bases are sticking out into the middle of this helix. And then they are pairing up. So adenine is pairing with thymine, guanine is pairing with cytosine. So our purines are pairing with our pyrimidines here, which makes all of these the same length. When our adenine pairs with our thymine and when our cytosine pairs with our guanine, those are the same length. So we have the same width apart for these backbones, these helical backbones there. So we have 2 types of bonds that occur in DNA structure. So in this sugar phosphate backbone, we have covalent bonds. We talked about this back in chapter 2 for our types of bonds that we see in molecules. So between the sugars and the phosphates, we have these really strong covalent bonds. And then the bases, when they're pairing up in the middle, these are hydrogen bonds. So they are relatively weak on the scale of bonds, but they are going to be holding these 2 chains of nucleotides together. So the way that these helical structures form the double helix is through hydrogen bonds between the purines and the pyrimidines. And this is just a little bit more detailed photo, or not photo, but image of DNA structure. And I wanted to point this out because it shows these hydrogen bonds between the bases. So between adenine and thymine, we have 2 hydrogen bonds. And then between cytosine and guanine, we have 3 hydrogen bonds that are occurring there. And then I also wanted to show this figure because I wanted to point out these parts of the molecule. This is called the 5' end and the 3' end. And this is important because these 2 helical structures that form the double helix are basically opposite in orientation. So, they read in one direction on one side, and then the opposite is happening on the other side. So you can kind of see how the phosphate group is attached to this part of the sugar, and then if you flip over here to the other side of the DNA molecule, it's opposite, right? It's going the other direction. So 5' and 3' refer to the number of the carbon in the molecule. So within our deoxyribose sugar, this carbon right here that's attached to the nitrogen of guanine, that is our first carbon. When you go down here to this part of the structure, that's our second carbon. This part here is our third carbon, so that's why it's called the 3' carbon. This one is the 4th one, and then this little bend right here coming off of our this sort of 5-sided structure and then going up to our phosphate group, this bend, that line right there, that's our 5th carbon. So that's our 5 prime. That's where our phosphate group is attached. So we have our 5 prime to 3 prime end on this side, and then our 5 prime to 3 prime end going the opposite direction on the other side. So this is going to be really important when we start talking about how new nucleotides are added to the growing strand that's being replicated, because certain enzymes can only proceed in certain directions, in certain, like from 5 prime to the 3 prime end. So we want to make sure we understand what those numbers mean, 5 prime and 3 prime, and how that plays into replication. So the first enzyme that's involved in DNA replication is an enzyme called helicase. So this is basically unwinding or unzipping the 2 strands of DNA because they are bound together. So we need to separate them in order to get in there and read those nucleotides and build a new strand. So it's going to break those hydrogen bonds that are holding the nucleotides together, and it's going to separate the strands so that our next enzyme, DNA polymerase, can get in and make a copy of the DNA. So DNA polymerase, that's what's building our new DNA. It's reading our original DNA strand and it's building a complementary strand. And it can only add new nucleotides to the 3 prime end of the growing molecule. So it has to go in the 5 prime to 3 prime direction. So because of that, we can go in one continuous motion on one side of the molecule, right? Because we can, if we're building in 5 prime to 3 prime direction, we can read from the 3 prime to the 5 prime continuously in one strand. But in the other strand, because that's opposite, it's going the opposite direction, we kind of have to do it in little chunks. So we're going to break this down in a bit more detail on another slide, but basically, we have one side of our DNA molecule that's replicated continuously, and we have the other side of our DNA molecule that's replicated in pieces, and then we have to piece all of those pieces together. Within our replication, we're going to end up producing 2 identical copies, so they have the identical sequence of nucleotide bases. They're exactly the same, and each DNA molecule has one old strand and one new strand. So instead of going in and making a copy of our DNA and then having one completely old copy and one completely new copy, we have our 2 copies have one old strand and one new strand. And this is referred to as the semi-conservative replication of DNA. So we have one old strand, one new strand. That's the semi-conservative DNA replication model. DNA replication doesn't just happen at the beginning of the strand, and it doesn't just start at the top and work its way to the bottom of this linear piece of DNA. It starts at something called the origin. So the origin is just this part of DNA that codes for the beginning of replication to happen. So it starts kind of in the middle and makes this bubble, and there are multiple origins happening on the linear piece of DNA. So as these bubbles get bigger and bigger, they run into each other, and then it forms those fully complete linear pieces of DNA. So we've got multiple origins, multiple bubbles happening on our pieces of DNA. Eventually, we're going to get one complete linear strand for each of our DNA strands there. So this is the more complicated breakdown of DNA replication that I want to talk about. I think this is a really good image to focus on if you want to study for how DNA replication is happening. Because if you understand all of the words on this image, you're going to understand how DNA replication is happening. So within our strand of DNA, this is happening in the origin. So this is at one of our origins of replication. So this is going to be one of those bubbles that's growing. So these are the things we've already talked about, the enzymes we've already talked about. These triangles here are helicase. So these are the enzymes that are unzipping our DNA, unwinding it so that we can have access to these nucleotide sequences. We're breaking down those hydrogen bonds between the bases to separate out the 2 strands. And then we've got one of those on both ends. So one on this side of our origin, one on that side of our origin. So it's expanding in both directions. And then we also have our DNA polymerase. So this yellow rectangle there, that's our DNA polymerase. And our DNA polymerase is the enzyme that is building nucleotides, adding nucleotides onto an existing strand. So things on this image that we haven't quite talked about yet, this is one of them. So this is the leading strand. So our DNA polymerase is going in the 3' to 5' direction. So you can see this is the 3' end of our DNA. This is the 5' end of our DNA. This strand here is referred to as the leading strand because this is just all one continuous piece of replication here. So when we're replicating in the 3' to 5' direction, the strand that we are building is starting at a 5' end, and we're adding nucleotides to the 3' end of that growing strand. Okay, we're always going to be adding nucleotides to the 3' end of that growing strand. And this one can just keep going and keep building on this side. DNA polymerase can only add on nucleotides if it has a nucleotide to add on to. It can't just, out of nowhere start adding nucleotides. So we have to have this little RNA primer. This is something that gets taken off and replaced with DNA after we're done. But in order to start adding DNA nucleotides, we have to have this little RNA primer. So anytime we're starting a piece of replication, we are going to have an RNA primer there. So our leading strand, we have one continuous addition of nucleotides, adding nucleotides onto that 3' end of our leading strand. The other strand we have is called the lagging strand. So this is the one that we have to kind of replicate in pieces. So we still have our DNA polymerase here, but we have multiple DNA polymerases that are working because we have to start replication in multiple spots. So again, we are doing this because we can only add nucleotides to this 3' end. So we can't just start on this side and work in this direction. We have to start over here on the left and work our way to the right. So we've got to do it in chunks. So these little chunks that are created, these are called Okazaki fragments. So named after just the guy who was involved in their discovery. So Okazaki fragments there. And then this enzyme here, DNA ligase, that's coming through and it's taking these Okazaki fragments and it's merging them together. So taking these little chunks and putting them into one strand. So that's what's happening in our lagging strand over here. We've got little pieces that are being replicated. These little pieces are called Okazaki fragments. And then DNA ligase comes in, takes those Okazaki fragments and merges them together to create one strand of DNA. So if you go through this image, and this image is in your textbook, it is in your textbook, figure 9.10. So you can take a look at that, make sure you understand all the pieces of this that are going on. So let's move on from replication and go to transcription. So transcription, we are going from DNA to RNA. So we're still within our nucleic acid macromolecule here, but we are transcribing what is in our DNA and putting it into RNA. This is something that's happening in the nucleus, right? That's where our DNA is housed. So that's where we're going to be transcribing this in the nucleus. Instead of using DNA polymerase, right, we don't need to add DNA nucleotides, we need to add RNA nucleotides. So our enzyme there is going to be RNA polymerase. And again, DNA replication is not required for this to happen. We always talk about replication when we're talking about transcription because the process is very similar, but we don't have to replicate just to transcribe, right? Transcription is happening a lot because we need this RNA to create proteins for cellular processes, but we don't have to replicate if we're going to transcribe. We only need to replicate if we're going to divide. So this is just comparing the structure of the sugars in RNA and DNA. So in DNA, we have our deoxyribose sugar, and in RNA, we have our ribose sugar. So you can see the carbons are labeled here. This might help you if you were a little confused with how the carbons are labeled when I was explaining it before. So our 1' carbon over here on the right side, and then kind of as we go clockwise around our sugar, we go to our 2, 3, 4, and 5' carbons there. And then the difference between ribose and deoxyribose is that deoxyribose is missing an oxygen, right? Deoxy, it's missing an oxygen, and it's highlighted here in red. It's the 2' carbon there. In ribose, we have an oxygen and a hydrogen that are attached, but in deoxyribose, we're missing that oxygen. Okay, so that's why it's called deoxyribose. So the sugar here is different in DNA and RNA, but other than that, very, very similar structure. We're just missing an oxygen in our deoxyribose. In addition to the sugar, there are a few other differences between DNA and RNA that you're going to need to be familiar with. So we've got our sugar highlighted again up here on the first line. The second line here is referring to the nucleotide bases that DNA and RNA have. So DNA, we talked about the purines and the pyrimidines. So our purines are adenine and guanine, and our pyrimidines, cytosine and thymine. RNA has a lot of those as well. It has adenine, it has guanine, it has cytosine, but RNA does not have thymine. It has a base called uracil. So it's still that single ring structure, but we never have thymine in RNA. We only have uracil. So when we are transcribing this, it's going to look a little different than when we are replicating, right? We are making complementary strands with our thymine in DNA, but again, there's no thymine in RNA. We have uracil instead. DNA is double-stranded, so our form there, our double helix structure. And then RNA, this is generally a one-stranded molecule. Really the only time in biology that I can think of right now that you would see double-stranded RNA is in some viruses. You can get double-stranded RNA, but that's really the only example I can think of. Generally, this is a single-stranded molecule. So as far as the functions are concerned, our DNA is storing this information, and then our RNA is carrying the information. So it's taking it from our DNA, bringing it out of the nucleus into the cell so that we can make proteins. So our sugar is different, our nucleotide bases are different, our form is different, and our function is different. So we're going to make sure that we understand the differences between DNA and RNA. When we talk about the actual process of transcription, that's occurring in 3 steps. So transcription is going to be initiated. We are going to elongate our RNA molecule. So we're going to build our RNA molecule, and then we're going to terminate transcription. So we're going to stop it. And these are all involving RNA polymerase. We're going to bind to the DNA to initiate transcription, aided by some proteins here called transcription factors. Then we're going to add nucleotides to our growing molecule, and then we're going to stop copying the gene. So we stop copying because there are sequences of DNA called terminators or terminating sequences that cause RNA polymerase to stop. So the DNA says, we're done, we don't need to copy anymore. Because when we were talking about DNA replication, we were replicating everything. When we're talking about transcription, we're just transcribing a small piece of the DNA. Our RNA is going in and transcribing because we just need to make this certain type of protein. So we don't need to copy everything. We just need to start it. We need to copy what we need, and then we need to stop it and get out of there and make our protein. So we don't need to copy everything or transcribe everything, rather. So when we do our initiation, when we look at initiation, our DNA is just going to partially unwind, so forming one of those bubbles, just like we saw in our origin of replication, and we're going to form a transcription bubble. We know where to start because there are sequences of DNA called promoters. This promoter region is telling the cell what needs to be transcribed. And then the actual molecule that's involved in opening the DNA is going to be our RNA polymerase. So in DNA replication, we had helicase that was unwinding our DNA and DNA polymerase that was building it. But RNA polymerase can actually go in here and open up the DNA in addition to working to add those nucleotides. So for elongation, we are transcribing from what's called the template strand. So if you look down here in this image, we have one strand of DNA that is our template, and then we have the other strand that's called our non-template strand. So our new RNA molecule, remember, we are always building, we're adding nucleotides onto this 3' end. So we are building in a 5' to 3' direction here. This is going to be almost identical to our non-template strand, which makes sense, right? Because we are, adding these nucleotides, these bases that are pairing up with our template strand. It's going to look a lot like our non-template strand, but you can see down here that, instead of having, so up here we have 2 T's that are bonded to these 2 A's. In our new RNA strand, we have 2 U's, right? Because we don't have T's, we don't have thymines in our RNA, we just have uracils. So this is going to look very similar, except instead of T's, we have U's. So our RNA polymerase is building this complementary RNA in the 5' to 3' direction, so always adding onto the 3' end. We can't add bases onto the 5' end. We have to add them onto the 3' end. And then another thing that's different comparing this to replication, when we are replicating, we're getting the formation of hydrogen bonds between these bases. We don't have that happening here. These bases are not going to be attached to each other, which is helpful because we don't want them to be attached, right? We want this RNA strand to go away, move out of the nucleus and into the cell to form these proteins. We don't actually want them to be attached. And then once this is all transcribed, we are going to be terminating this transcription sequence. So a termination signal is needed to stop it. So this is basically a series of repeated nucleotide sequences in DNA that tells RNA polymerase to leave and close the double helix of DNA, and then our new RNA molecule is released. Once it's released, it's going to go out into the cell, and then we're going to get our last part of this central dogma, which is translation, to get our actual proteins that are going to be created. So before we do that, before we head out of the nucleus and into the rest of the cell, we have to process our RNA a little bit. So when we just copy our RNA, we get sort of the series of what are called exons and introns, and we need to modify this a little bit to make it ready for translation and ready to be out in the big part of the cell, not just in the nucleus. So this is something that's only happening in eukaryotes, and it's happening in the nucleus in between transcription and translation. And there are 3 processes that happen within RNA processing. So you can kind of see it in this image, but we're going to break it down also. When you compare this top line to this bottom line here, we have this 5' cap that's been added. We have what's called a poly A tail that's been added to the 3' end, and then all of our introns have been cut out. So the first thing that's going to happen is we are going to add this 5' cap. So we need to add a cap onto the 5' end to make sure that the mRNA, and we're going to talk about mRNA, what that means specifically in a couple slides, but that's the type of RNA that's created in transcription. If we don't add a cap to the 5' end, it could be degraded by what are called exonucleases. It's an enzyme that floats around the cell that breaks down nucleic acids. Because once we have our protein made, we don't need this nucleic acid anymore, this RNA anymore, so it's going to be degraded. We don't want it degraded before we make our protein. So we have to add this 5' cap on to the 5' end. So we have a 5' to 5' bond that's formed here, so that we end up having a 3' carbon on the end of our 5' end of RNA. Okay, so that's going to make sure that it doesn't get degraded. So we have a cap added to our 5' end, so it's called a 5' cap. We also have the addition of a poly A tail. So the A's are just referring to the type of nitrogenous base that's on these nucleotides, so lots of adenines that are being added on to the back of our RNA. So we're going to have lots of A nucleotides added to the 3' end. This is just another way that we are protecting our RNA from degrading really rapidly. So you can see here, there's about 250 of these nucleotides that are added to the end of our mRNA sequence. So this poly A tail, so lots of A's on the 3' end. And then the last thing we're going to do is we're going to splice. So we are going to remove what are called introns, and we are going to leave behind what are called exons. So the introns are just sequences of this RNA that don't actually encode for functional proteins. So we want to take all of those out, and we only want to have the sequences that are encoding for functional proteins. So we have exons here, and then our introns that don't encode for anything. We will splice our RNA molecule to take these introns out, and then we are left with what's called our mature mRNA. So we've got our 5' cap, our poly A tail, and all of our exons here right in a row. So to overview, just an overview of RNA processing, we've got these exons and these introns. 5', this is referred to as an untranslated region. 3' untranslated region, because this is not encoding for anything there. It's just sort of indicating the beginning of what we need to translate into a protein and the end of what we need to translate into a protein, telling it to start and stop. But those are untranslated regions there, that UTR. And then when we're done processing this, we've got our 5' cap, we've got our poly A tail, and we've got that big region right in the middle that's going to encode for our functional protein. So the types of RNA that we're going to see, and this is important to understand for translation for our next step, because all of these types of RNA do something different. So messenger RNA, mRNA, that's what we've been talking about as far as transcription goes. This is what's carrying the genetic information from our DNA, from our nucleus, into the rest of our cell, to our ribosomes. We also have transfer RNA. This is called tRNA. This is what's going to be connected to our amino acids. It's going to take our amino acids from the cell, bring them to the ribosome so that we can build amino acids and make chains to form our proteins. And then we have rRNA, or ribosomal RNA. This is just working with proteins to actually form the ribosomes, which are going to be the structure that is involved in translation. So ribosomes can just be free in the cell. They could be polyribosomes, which are just clusters of ribosomes, or they could be attached to the endoplasmic reticulum. Remember, we have two types of endoplasmic reticulum. We've got smooth and rough. This is going to be the rough ER that has ribosomes attached to it. So our RNA is just forming the ribosomes with proteins. So let's talk about translation now. So the last step here in our central dogma of molecular biology, this is where we're going from RNA to proteins. So we're going from a nucleic acid macromolecule to a protein macromolecule. So this is translating the information from one type to another type of macromolecule. Our transcription, that occurred in the nucleus. This is occurring in the cytoplasm. So translation is happening in the cytoplasm. It requires the ribosome, so we're going to have that rRNA in our ribosomes, and it requires tRNA, so our transfer RNA bringing our amino acids into the ribosome, and this is going to follow transcription. So when we talk about what our ribosome looks like, again, this is what's involved in translation, so we need to know sort of how it's oriented here. We have two subunits in our ribosome. We have a small subunit, and we have a large subunit, so two subunits in this ribosome. Within the small subunit, that's what's going to hold on to our messenger RNA. So the mRNA that we made in transcription is going to be anchored in the small subunit of our ribosome. In our large subunit, we've got 3 sites, basically 3 slots where things can sit. We have what's called the A site, the acceptor site. So that's over here on the right. We've got our peptidyl site, our P site, there in the middle. And then we have our exit site, E, over here on the left-hand side. So we've got 3 slots here, basically. And these are going to be for our transfer RNA to come in, and they're going to sit in either of these spots, depending on what they need to be doing in translation. So the tRNAs are going to enter through the A site. So our tRNAs, again, are attached to our amino acids. They're going to be floating around in the cytoplasm. They're going to enter the large subunit and anchor here in the A site. Once they've anchored here in the A site, they're going to move over to the P site. And then we are going to have a new tRNA come in and sit in the A site. And then once we have these two sites occupied, each of the tRNAs that are sitting in here have an amino acid attached to them. So those two amino acids can form a bond between each other, and then we start to have this growing chain of amino acids. Once the tRNA has donated its amino acid to the chain, it moves over here to the E site and it exits. Okay? So this is really hard to demonstrate just me talking to you. So I do have a video that we're going to watch here in a couple slides. I don't know if it's actually going to work as far as this WebEx goes. So we'll try it. And then if it doesn't work, I'll post a link to it in the D2L site for chapter 9 so that you can watch it on your own, but we'll at least try it. I have no idea if it's going to work. So this translation occurs in 3 steps, just like transcription occurred in 3 steps. So we have initiation, where we have the ribosome assembling onto the mRNA, right? That strand of RNA from transcription is going to be anchored in the small subunit, and then we have the large subunit coming in and attaching as well. Elongation happens where the ribosome builds a protein. If we think back to our protein structure, we break it down all the way to that primary structure of a protein. It's just a chain of amino acids. So the whole point of what we're doing here is just to build a chain of amino acids. That's what elongation is doing here. So we're just adding one amino acid to the chain as we go through our mRNA sequence. And then eventually our translation will be terminated. So there will be basically a part of the RNA, that mRNA that's anchored in that small subunit that says, stop, we're done. We don't need any more amino acids. And once it reads that piece of information, everything just kind of disassembles and falls apart. Our ribosome falls apart, our mRNA is released, our chain of amino acids is released, and it can sort of assemble and bond on itself to form the more complex protein structure. So this stop sequence is referred to as a codon. All of the amino acids are basically built based off of what's called a codon. So a codon is a sequence of 3 nucleotides that codes for a specific amino acid. So this chart here is how we know which sequence of nucleotides is going to code for which amino acid. So to start translation, we are always going to see a sequence of 3 nucleotides that read A, U, and G. That is the sequence of nucleotides that tells everything that we need to start coding for a protein. And then our next 3 nucleotides, if we maybe have a C and then a U and then another U, that's going to code for an amino acid called leucine. So we might see that, and then maybe our next 3 amino acids are going to be U, G, and U. So that's going to code for a different amino acid, and then we're going to code for all of these amino acids in chunks of 3, these codons, and then eventually we're going to get to one of these 3 series of nucleotides that tells us to stop. We're either going to get a U, an A, and an A, a U, an A, and a G, or a U, a G, and an A. Once we get there, that doesn't code for an amino acid. It tells everything to stop, and then everything disassembles, and we've got our chain of amino acids. Okay? So this is the video. Again, we're going to try and watch this. I don't know how well it's going to play on your end, so we'll see how it works, and then I'll post it for you guys in D2L also. So we've got our DNA that's transcribed into our mRNA, and then we want to translate that into a protein. So we've got our ribosome there. mRNA is entering on the small subunit. We have a tRNA coming in and anchoring to one of our codons down there. Our tRNA is attached to an amino acid. We've got one in the P site and one in the A site. Once we have them, that's our anticodon there. That is a complementary piece of information to our codon, right? Our series of 3 nucleotides there. So that's how we know which one codes for which one. So once we have those matching up and the tRNA comes in, those amino acids are going to join together. Our used tRNA goes into the E site, the exit site, that no longer has an amino acid, so that one's going to leave. We don't need that one anymore. And then our P site has this growing chain of amino acids. We have a new codon in our A site, so that's going to code for a new amino acid. That tRNA is going to come in, dock right there in the A site. Then it will move over. Our bond is formed between our amino acids. And so you can see this just keeps going and going and going. We've got this chain of amino acids that's growing. We've got them entering in the A site, leaving in the E site, and just have this long chain of amino acids that's created. So that's kind of what's happening here in our ribosome. And then eventually we're going to get to a stop codon that tells everything to fall apart. Now we've got our chain of amino acids that can form our protein. Okay. So we're going to make a protein. We're going to talk through this transcription and translation, starting with DNA, so that we can get our chain of amino acids. So this is something that you might be presented with in the chapter quiz, making a protein or a chain of amino acids from a piece of DNA. So the first thing I would recommend you do if you're given a piece of DNA is to split it into sets of 3. This is going to make it easier just to not only just keep track of the letters and make sure you haven't missed any letters, but this is also going to be important for when we're looking at those codons and which amino acids they code for. This is just going to make it easier. So I'd recommend just break it into sets of 3 right off the bat. And then we're going to make our RNA. So again, these are going to be complementary strands, but we don't have thymine, we just have uracil. So T always pairs with A, so we're going to have an A there. When we're talking about DNA, A pairs with T, but RNA doesn't have T, that has uracil, so that's a U. And C always pairs with G, so we're going to have C and a G there. So this next one, we have CGC. When we're talking about our RNA, that is going to just be GCG, right? Our G's and our C's aren't any different. Those are the same in RNA and DNA. Our next set of 3 here are 3 A's. A's always pair with T's. We don't have T's in RNA, so that's going to be 3 U's there. And then in DNA, this section here, we've got T, C, and C. T always pairs with A. C always pairs with G, so we're going to have AGG. And then our last set of 3 here, we've got an A, a T, and a C. So A always pairs with T, but we don't have T's in RNA, we have U's. So we're going to have U, A, and G. So this is our complementary RNA strand for our protein. And now what we do is we're going to take this chart and we're going to figure out which amino acids we're coding for here. So the first letter is this side, this vertical side here. And then the second letter goes across the top. So you can just kind of track down what series of amino acids you're looking for. So the first one we're looking for is AUG. So the first letter is A, second letter is U. So we're going to go down to this box and we can see that AUG, this is our start codon. Start codon always codes for an amino acid called methionine. So that is referred to as MET. So you can just use the shortened version of the amino acid names here. Our next one, GCG. So first letter is G, second letter is C. GCG refers to amino acid ALA. So Baylor asked a question, how do we know what letter pairs with the RNA? I'm not sure what you mean here. So this is our DNA strand. In transcription, we make our RNA strand, and then this is the strand that is anchored in the small subunit. So this is what we're reading here. So that's what's referring to this chart. And then this sequence here, it codes for, like this AUG codes for methionine because this is the codon. So that codon is in that small subunit, and then the tRNAs that are floating around that are attached to the amino acids, that anticodon, they have the opposite letters. So the anticodon for this would be like a U UAC. So a UAC is going to be anchored to that amino acid. So that's how it's going to come in and basically sit in that large subunit. And then we're going to get that series of amino acids. So we are RNA pairs, the letters pair just like DNA does, except there's no T's, right? So A's pair with U's, U's pair with A's, and then G's and C's pair with each other. So when we transcribe our DNA into RNA, we get that, and then this mRNA is all we need to know to use this chart to figure out our amino acids. I'm not sure if that answered your question, Baylor, but okay, great. Okay, so our second series here, GCG, we're going to go to G and then to C. So we've got ALA for our second amino acid. UUU is our third series of 3 here. So we're going to go to U and then to U. So we find it here right up at the top. AGG is our next one. So first letter is A. Second letter is G, so we're going to go down here and find AGG down there on the bottom. And then our last one is UAG, so we're going to go to U over here for our first letter, A over here for our second letter, UAG is our stop codon. So it doesn't code for an amino acid, it just tells us that we're done. We don't need any more amino acids to make this protein, so we can just stop right here. And disassemble everything, and our amino acid chain is good to go. So these genes, we've talked about, DNA, RNA, and proteins, but these proteins are what are resulting in the phenotypes, right? In chapter 8, which you guys did before the break, we talked about genotypes and phenotypes and how genes correspond to physical traits. And that is happening through transcription and translation. So the information in DNA is transcribed into RNA, and then when it's translated into proteins, these actual proteins are what are coding for the phenotypes. So you can see here, we've got 2 mice. They have a very similar sequence of DNA, except we're just one base is different right here. So in panel A, we have a G. And then in panel B, we have an A, but everything else is the same. We've just got one nucleotide that's different. So when it's transcribed, we're going to get something that looks different in that one nucleotide base. And then when we have that one nucleotide base being different, we're getting a different amino acid that is coded for in our protein. And then when we have that different amino acid, we're actually getting a different phenotype. So these phenotypes, these physical traits, are products of the proteins. So when we have different series of nucleotides in our DNA, we get different series of amino acids in our proteins, and then we have different phenotypes. So this is all sort of related to what we talked about, or what you guys talked about in chapter 8. And then the last thing that I wanted to talk about is just mutations. So a mutation is when there is a change in the sequence of nucleotides in this DNA. So this could be maybe something happens in replication. Maybe there's some sort of mutagen present that's causing nucleotides to change. Like sometimes UV light can cause a mutation in DNA. So here's an example down here. This is a fruit fly. So there's a mutation in just one gene that causes them to develop legs instead of antennas, right? So here they have antennas, and here they have legs coming where their antennas should be. So these mutations are changing the nucleotide sequences, and they can result in pretty big changes in the phenotype, right? So there are 2 types of mutations. There are 2 general categories. The first category is a point mutation. So this is kind of what we saw when we were just talking about the differences in those 2 different colors of mice, where we have one nucleotide that is different. So a point mutation is a single nucleotide, a single base change that results in a change in the amino acid sequence. So we have our normal DNA here. This is what's corresponding to normal hemoglobin. So this is an example of sickle cell anemia here. So our normal hemoglobin, right here in this middle nucleotide, we have a T and an A. But in our mutant hemoglobin DNA that codes for sickle cell hemoglobin, Instead of having a thymine in the middle, we have an adenine, we have an A. So instead of coding for an A in our mRNA, we're coding for a U. So we have this one nucleotide that's different, which makes this one amino acid different. So it's just changing one amino acid there. The other type of mutation is called a frame shift mutation. So this is where we are adding or deleting a nucleotide from the sequence, and this can change everything in the future, right? Like if we add one nucleotide here to our sequence, we're not just changing this one amino acid, we're changing every amino acid after it, because we have inserted or deleted a nucleotide here. So this can have much larger impacts, a frame shift mutation, than a point mutation, because it's changing so many of the amino acids rather than just one amino acid. And even though these mutations are changing our DNA, that doesn't necessarily make them bad. Mutations are actually really important for creating different types of alleles. So alleles you guys talked about in chapter 8, where it's just different types of the same gene. And then when we have these different alleles that are created, that can help lead to just a diversity of genes. And this is one of the ways that evolution actually works. And we're going to be talking about evolution here pretty soon, probably on Wednesday. So this is one of the ways that evolution works, is on mutations. So even though mutations sound bad, and sometimes they are really bad, sometimes they can actually lead to things that are helpful, or at least just enriching the diversity of genes that we have. And diversity is super important for evolution. So it's not necessarily a bad thing. It can be bad, but it's not bad all the time. Okay, so that's all we've got for today. So if you haven't marked yourself present here in the chat, go ahead and make sure that you've at least said hi, or said that you're here in the chat, so I know to give you credit for being here. I'll post this video to D2L, and then I'm also going to post an announcement just to make sure you guys understand what we need to have done today, and what we need to have done for the rest of the week. So I will be here on Wednesday to do another lecture on Wednesday. So hopefully I see you guys then. Feel free to message me if you've got any questions, and hopefully you guys are all staying safe and healthy. All right, I'm going to go ahead and stop the recording here and end the meeting.